Hello and welcome back to my second part of the video, that is both transcription modification. In the first part of the video we have discussed about the polyadenylation and capping process. In this video we will be discussing about the splicing process in detail. In the last part of the video I have already told you why post transcriptional modification is needed, because the mRNA which is formed from the processing, that is from transcription, is mainly immature mRNA. It is needed to be certainly modified so that it is matured enough to form the protein. That's why certain modification is done to make the pre-mRNA into a matured mRNA. So let's begin the third part of the processing of the mRNA, that is splicing process. Before that, let's understand what is splicing. Splicing means to remove or to splice out. Okay. So you know that the mRNA consists of both introns and exon. If you've been wondering what is an intron and what is an exon, in simple word we can say exons are the coding part of the mRNA which is responsible for the helps in the protein synthesis, whereas intron are the non-coding part which do not take part in the protein synthesis. So it is very much essential to remove the introns so that only exons remain and a proper structure is formed in the mRNA which helps in the protein synthesis, and the intron, that is the extra part, is removed out which does not take part in the protein formation. Okay. As you all know that while the transcription of the RNA, both the intron and the exons will be formed and the introns will be removed out. The process by which the introns are removed out is known as splicing, is to remove. Or this splicing technique, you must be wondering that when does splicing occur. So as I've already told you, the first modification which takes place in the RNA is the capping process. Remember that the capping is the first process which takes place in the mRNA, pre-mRNA which is formed. First process is capping, second is the polyadenylation, and third step is the splicing step. So only after the 5' and 3' end processing only the splicing process is done. This is very important to be noted down, because sometimes in exam it is asked to arrange the sequence that which process, which modification takes place first. First is capping, then polyadenylation, then is the splicing technique. And capping, remember that the capping takes place co-transcriptionally, that is along with transcription the capping process will takes place. Okay. Next, why the splicing is needed? As I have already told you, splicing is needed to enable the eukaryote to increase their coding potential. What do you mean by this statement? Because if you remove the unnecessary things from the mRNA, it will be very convenient for the mRNA to process itself as well as to produce the protein structure. Otherwise it will be very difficult always to carry the uncoding part which is not at all taking part in any activities. For example, there is a group of people in a class and 5 of them are functioning, that is they are capable enough to do a task, and 5 of them are incapable, means they are not enough to do that task. Okay, 10 people. So in a competition, there will be a competition among these two group. Then what will happen? Obviously, as this 5-member group, that let's say group A is more competent enough than that of the group B for a certain task. So obviously this group A will go and take part in will move forward, or will be winning the task, you can say, and this group will be removing. In same things happen. Both intron and exon is a part of an RNA, but as the exon is a coding part and it will take part in the process of protein synthesis, that's why they are remaining and the introns are removing. In certain cases there is seen that in rare cases intron also take part in certain protein formation. We will not go into that zone because majority of introns are non-coding. Okay, exons are coding part. So now you might be clear about that, what are intron, what are exon, and why all is these things are necessary. Let's understand with a greater in detail, or you can say clearly with the help of this diagram. Okay. you might be seeing this, that is, this is a pre-mRNA, this is the exon 1. Okay, many exon and many intron will be present. Between exons, the introns are present. So this is exon 1, this is a intron, it is not clearly visible, but this is intron, exon 2, intron 2, then exon 3, and so on. It is a pre-mRNA. Why I am mentioning pre-mRNA? Because it is not yet matured, because the introns are already present. Okay. Then what happens? The spliceosome. What is a spliceosome? Spliceosome is an enzyme which is responsible for removing the intron. So spliceosome, we will be reading in the process method different types of splicing process in detail, but let's understand the overview. Okay, what happens in the splicing process? So what happened? The spliceosome will come and attached. This is a complex, okay, it come and attach to this intron part and result in the removal of intron by a formation of lariat, or that is a round like loop like structure, and the rest exon part will be joined. This is the spliced mRNA, or you can see the matured mRNA. This is the overall process how the that is the splicing takes place. Now we will be dealing with the types of splicing. Before that, let's understand that first what happens, 5' capping will take place. You can see we have discussed in the last part, and this is the coding sequence, which you can say this is the exon part, and this is the non-coding sequence, that is the intron part, and this is the poly A sequence. This is the second processing method. First processing method is capping, second processing method is polyadenylation, and third is the removal of this part, that is the non-coding part. You can see here that this coding sequence is responsible for the protein formation. Okay, this is the overview idea which I have been telling you till now. Now moving to the types of splicing. First is the exon skipping. What is exon skipping? First process is that as there are 3 exon, it is not always necessary that all the 3 exons will be joined. Sometimes what happens, one exon will be skipped and rest 2 exons will be joined to form a structure which forms a particular type of protein. Let's say for this exon 1 and exon 3 will combine and it will form a certain type of protein, and here exon 2 is skipped. That's why this process is called exon skipping. Let's say for certain example 2, exon 1 and exon 2 will be joined and exon 3 will be skipped. In other type, exon 2 and exon 3 will be joined and exon 1 will be removed. So you can see here by this process there will be a variety of proteins will be seen. So always it is not necessary if there are exons, here I have taken 3, there will be number of exons will be present. It's always not necessary that all the exons will join. Sometimes 1 exons will be skipped, sometimes 2 exons might be skipped. Okay, so this is one method of splicing which is known as exon skipping. Moving to the second part of the splicing process, that is cryptic splicing signal. What is cryptic? Suppose there are 2 exons are present, that is, let's say exon 1 and exon 2. What happens in this splicing, only a part of exon 2 will be joined to the exon 1. That's why it is called cryptic splicing selection. That is, a portion of exon 2, you can see here, not whole part of the exon 2 is not joined with the exon 1, only a part of it is joined here. Okay, this method is known as cryptic splicing signal. Okay, moving to the third part of the third type of splicing is the alternative splicing. What do you mean by alternative splicing? Why alternative splicing is formed? Because to form different types of protein. Because you know that every exon results in the formation or joining forms a one type of protein. Let's say what happens, this is a gene and these are the mRNA, that is exon 1, 2, 3, 4. Okay. What happens in alternative splicing? Suppose exon 1, 2 and 3 forms 3 joins and form protein A, and in the second type, that is 1, 2 and 4 joins and form protein B. Similarly 2, 3, 4 will join and form protein C. This is known as the alternative splicing. Okay, so why this alternative splicing? What is the role of this? It results in the formation of different types of protein. Next variety is trans-splicing. What is trans-splicing? The word trans means it comes from different chromosome. Okay, suppose this is the chromosome 3 of one consists of one exon. Okay, this is the exon of chromosome 3 and this is the exon of chromosome 2. So trans-splicing results in the joining of exon from the two different types of chromosome. So that's why this is called as trans-splicing method. So these are the major types of the slicing, that is exon skipping, cryptic splicing techniques, alternative splicing and trans-splicing. Now comes to the method how the splicing takes place. Okay, now we will be dealing with the exactly what is happening. Okay, what happens? So first let's understand the structure first. This is the pre-mRNA. Okay, let's understand. Wait 1 minute. Okay, this is a pre-mRNA. Okay, the pre-mRNA has 5' end and 3' end. You can see here. This is exon 1 and this is exon 2 and the middle part is known as the intron. The intron, the middle part of the intron has a branch point. Okay, branch point that is known as the adenine. The branch point consists of the adenine which has a 2' OH end. You know the adenine structure has an OH structure because we have dealt with the nitrogenous bases already. Now by now you might be clear that it has a 2' OH end, and this exon 1 has GU sequence. You remember this sequence because this is very much important. GU sequence is present in the exon 1, this region, okay, and this consists of AG sequence, okay, and this site is known as the 5' splice site, okay. This sequence is known as 5' splice site and this region is known as 3' splice site, okay. This region is known as 3' splice site. What happens in this technique? This branch point, that is the intron, contains a 2' OH end. This 2' OH end will form a nucleophilic attack on the 5' splice site. As a result, what will happen? This portion will be removed. Okay, this much portion will be removed and will be attached to this end. You can see here this region, that is PA, PGPU. Okay, up to this much portion will be a splice attack will be done right here, as a result of which there will be a breakage and they will attach here, and this 5' splice, this 5' end will form a OH end. This OH end will again form a attack, nucleophilic attack on this 3' splice site. As a result, what will happen? This much portion will be removed and the intron will be removed. So our motif is complete. Now the intron will be removed in the form of lariat and the spliced exon, that is the 5' PN, 3' that is the exons will be added. Okay. So now let's understand the theory. So the 2' OH, a branch point adenine, attacks the phosphoryl group of the G in this 5' splice site, as I have already told you. The attack leads to the breaking of phosphodiester bond between the 3' end of the exon and the 5' end of the intron. As you know that there are phosphodiester bonds will be present, so this bond will be broken down. As a result of which OH will be formed. Formation of new phosphodiester bond between the branch point A and 5' end of the intron. Okay, there will be a new bonding will be taking place here, and this OH will form again a nucleophilic attack on this 3'. As a result, this will break the bond and the intron will be removed in the form of lariat. You can see here, and the exons are joined. So this is the splicing method, how the splicing is done. Okay. So in this part of the video, let's keep it this much only, because the next part of the video is little bit there are many more to be discussed. Okay, so in this we will do this in a second part, because this much will be, I think this will take some time. I don't want this video to be lengthy because it's very important, so part by part it should be understood. So let's keep this much today. In the second part, we will discussing about other 3 types of the splicing technique. So before that, I request you to first understand what is transcription, how transcription taking place in prokaryote, eukaryote, how the modification has taken place, that is capping, then polyadenylation, okay. Then this splicing I have discussed only half, and rest half we will be discussing in the next part of the video. Second most important thing, molecular biology is very important part and crucial part to be understood. Once you get through it, everything will be cleared. So I request you all to watch the video series I have made already. share as much as you can, because these are very important things. In BSc, also in MSc, it's and entrance point of view is also asked many time. So please go through it. If you have any doubt, to ask in the comment section. Subscribe to my channel if you are new, and share as much as you can. So that's all for today. Thank you and have a nice day.